Hi, my name is Snigdha, and I chose to research about the tumor microenvironment, which is the environment surrounding a tumor, including immune cells, blood vessels, and signaling molecules like TGF-beta 1, which is an immunoregulating protein. The tumor and the microenvironment interact very closely, so I chose to research about TGF-beta 1. I chose to research this topic because I've always found it interesting how you can make so many connections at a molecular level. So let's get into some background research. The TGF-beta superfamily consists of growth differentiation factors, which function in cellular development, bone morphogenic proteins, which regulate bone maintenance and repair, anti-Mullerian hormones, which function in reproductive development, and the TGF-beta family. TGF-beta is a ligand that regulates an organism's immunity by binding to TGF-beta receptors. A ligand is a molecule that binds to a receptor, which then activates a cellular signaling pathway. A ligand is like a message sent from one cell to signal another. During these processes, SMAD proteins act as the main receptors for TGF-beta. TGF-beta signaling pathways assist in cellular development and growth. Additionally, TGF-beta 1 plays a critical role in wound healing, immunoregulation, and the maintenance of cell growth. However, mutations in TGF-beta 1 in the tumor microenvironment can cause it to function differently, and instead, TGF-beta 1 can help the tumor grow, playing a critical role in uncontrolled cell proliferation. Additionally, TGF-beta 1 can supply the tumor with nutrients and oxygen, which is known as angiogenesis. So, to summarize, TGF-beta 1 can play a critical role in one's immunity. However, with the dysregulation of this tumor-suppressing ligands, TGF-beta 1 is no longer an anti-proliferative protein, and instead, TGF-beta 1 acts as an oncogenic factor promoting angiogenesis, metastasis, and tumorigenesis, therefore allowing the tumor growth. Throughout my research, I explored the effects of mutations on TGF-beta 1. I also used publicly available amino acids obtained through NCBI. Then I create an alignment in benchling and compare them with known mutations that researchers have found. So first, I collect a list of mammals through NCBI. Then I search the name of the animals in an age database to identify their average lifespans. Then I note them down in a spreadsheet and order them from shortest to longest lifespans. I do this because short-living species are a proxy for cancer, because species with lower lifespans have a higher rate of mutations related to cancer. Then I create a sequence in benchling, keeping the same order of mammals. I then use a sequence map to find areas with possible mutations, indicated by the red bars. Finally, I compare the mutations I found in the alignment with mutations in COSMIC and ClinVar, which show me mutations that researchers have found from. So before I begin and present my results, I will first explain conservative and non-conservative mutations, which I will use to analyze my results. Conservative mutations won't change the function of the protein. So the original amino acid and the mutated amino acid would have the same characteristic, so same charge and same polarity. However, non-conservative mutations are mutations that will drastically change the function of the protein. So the original amino acid and the mutated amino acid would have different characteristics, so different charge or different polarity. The first mutation I found was at position 162, where the golden hamster and house mouse have a serine, while the rest of the species have a leucine. This is non-conservative because serine is polar, while leucine is nonpolar, so different characteristics. The next mutation I found was at position 212, where the golden hamster, house mouse, guinea pig, and pig had a glutamic or aspartic acid, while the rest had a glycine. This is non-conservative because glutamic acid and aspartic acid are polar, while glycine is nonpolar. The next mutation I found was at position 16. The golden hamster and house mouse had a proline, while the rest had a leucine. This is a conservative change because both proline and leucine are both uncharged and nonpolar. The next mutation I found was at position 148. The golden hamster and house mouse had a proline, while the rest had a valine, except for the guinea pig, horse, and African savanna elephant. This is a conservative change because both valine and proline are nonpolar and uncharged. Finally, the last mutation I found was at position 238 to 239. The golden hamster, house mouse, and guinea pig had an isoleucine, while the rest had a glycine. This is a conservative change because both isoleucine and glycine are nonpolar and uncharged. So, same characteristics. Therefore, the mutations that were conservative were positions 238 to 239, positions 148, and position 16. While the non-conservative mutations were position 162 and position 212. After looking through the same mutations through ClinVar and Cosmic, I found them in positions 212, 238, and 148. The mutation at position 212 was a mutation from glycine to tryptophan. However, the one I had found was from glycine to glutamic acid. The next mutation I found in Cosmic was at position 148. However, the one that they had found was valine in methionine, but the one I had found was valine in proline. The last mutation I found in Cosmic was at position 238, where there was a mutation from glycine to valine. However, the one I had found was glycine and isoleucine. Therefore, there were some similarities between the mutations I found in my alignment with mutations that researchers have found of existing mutations in TGF-beta 1. Therefore, using the data and analysis, I identified the most promising sites. The most promising site was position 212 because it was a non-conservative mutation. Additionally, my mutation was also seen in Cosmic, and the mutation in Cosmic and the mutation I had found shared a glycine. The next most relevant site is position 148, because although it was conservative, it was also seen in Cosmic. And the mutation in Cosmic and the mutation I had found both shared a valine. The next most relevant site is position 238 to 239, where although it was conservative, the mutation was also seen in Cosmic, and both mutations shared a glycine. Finally, position 162 could be relevant because it was non-conservative, which means it could be a driving factor that changed the function of TGF-beta 1 to cause it to be proto-oncogenic. Therefore, the mutations I had found could be really relevant for further research because they were closely related with known mutations of TGF-beta 1. Additionally, some were non-conservative, so they could be the reason mutated TGF-beta 1 can help tumors grow rather than being a tumor suppressing factor. During this project, I have learned the process of research, and my results can be applied to future cancer research because looking for possible TGF-beta mutations ahead of time could be significant in stopping further problems like excessive tumor growth. Thank you.
Thank you, Snigdha. Very cool. So, general question from the audience. What was obviously lots of interesting insights from your research. What was the most surprising to you after completing this work?
I think the most surprising thing for my whole research part was after going through the challenges of, analyzing my data that I had found and figuring out what is relevant and what isn't, and then comparing it to known mutations, I found it very surprising that some of the mutations were actually there. They actually existed. And it also helped me build confidence really that the ones I had found were also in Cosmic and Clinvar, and it could actually be relevant to further research. And yeah, that kind of helped me with my whole research.
It's so cool. Yeah, no, it all kind of fits together when you start looking into it. Very cool.
Yeah.
Thank you so much.